All right, well, welcome back from lunch. My name is Jennifer McPartland. I'm a senior scientist in the health program at Environmental Defense Fund, and it's my pleasure to orchestrate this portion of the meeting. This afternoon, we're going to kick it off with a couple of case studies. Our first case study will be from Dr. Tara Sabo-Attwood, who is an associate professor in environmental toxicology with the University of Florida. She will be describing work that she's doing looking at in vitro methods that are being used to explore potential toxicity associated with wastewater effluent. And I won't take any more of her time and just hand it to Dr. Attwood.
Hi, hopefully everybody can hear me okay, and that my presentation is up. I can't quite see, but sorry I couldn't be there in person, but I had some extenuating circumstances this weekend to deal with, but thanks to the organizers for allowing me to do this and still participate remotely. I've been asked today to talk about the state of the science of in vitro assays in the context of their utility in water monitoring programs. And so I'm going to do that on behalf of myself and Dr. Nancy Denslow, who couldn't be here today, but really sort of the expert in this area. I already mentioned today's talk is going to focus on aquatic scenarios. I'll spend some time introducing some of the bioanalytical assays that are being used specifically in water assessment scenarios. Talk about two case studies. The first will quantify bioanalytical assay responses to variably treated wastewater samples. And the second is going to talk about linking in vitro responses with in vitro effects. And these case studies will serve to lead us to challenges and forward thinking related to how in vitro assays may be integrated into tiered monitoring regimes and what gaps exist in our understanding of in vitro and vivo linkages with respect to complex samples. I'll also highlight forward momentum in the science as aspects to consider for discussion and consideration of how to relay and incorporate such ideas into the decision-making process. Because I'm a basic scientist and do not work in the regulatory realm, and we don't have the same toxicity database for complex samples, and most importantly, because my laptop was stolen this weekend and I had one day to re-pull this talk together, my talk is going to be a bit more data heavy, but the point is to use the data to highlight what we perhaps need to be thinking about. And so just to get started, Stan and others have already made a reference to adverse outcome pathways, which serve to represent linkages of knowledge based on structural biological events. And just to sort of reiterate a little bit that the both the molecular initiating events and the adverse outcomes serve as anchors of the pathway, with several key events sort of spanning between and linking these anchors. And so, as mentioned, select AOPs are currently directing prioritization of Tox21 and EDSP screening programs for individual chemicals. The in vitro assays I'm going to refer to are high throughput screening cell-based and cell-free assays that tend to be built around a lot of these molecular initiating events, such as receptor-ligand interactions, and they are used to elucidate MOAs, prioritize chemical testing, and aid in development of predictive modeling. In the monitoring and risk assessment fields, they are not necessarily meant to replace in vivo testing, but rather to provide information on chemical screening and prioritization, perhaps having a place in tiered regulatory frameworks to reduce more extensive testing practices. Over the last 5 years, these AOPs have evolved as new information is learned. Some of this was alluded to today, such as inclusion of metabolism and the value of quantitative chemical measures in support of predictive computational models. There's also been a movement towards utilizing in vitro assays to test complex environmental samples. I'm going to first present a case for using such assays to test wastewater effluent samples. Excuse me. The first study is published within the last year or two by Escher et al. And Nancy Denslow was involved in this, where they tested using a round-robin design several types of cell-based bioassays to ascertain their suitability, to benchmark water quality, and to assess efficacy of water treatment processes. They looked at 103 unique bioassays that are being used in ToxCast or Tox21 screening. One assay available through Adagene as just an example enables quantitative assessment of 73 nuclear receptors and transcription factors, and their associated activities simultaneously in cells. And they target things such as the aryl hydrocarbon receptor, estrogen receptor, which has already been talked about today, and things like retinol gas receptor. And so if you look at these assays in response to a complex wastewater treatment effluent, out of 73 targets, they saw that there were 23 total hits, 11 that had what we're calling high activity, which is shown here in this table. And just to note, the level of activity, although depicted as pluses here, were assigned from quantitative EC10 and EC50 values. But in their round-robin approach, they use several other assays, such as the gene laser assay, which really looks at receptor activation on an individual level. And this is just really showing what type of data you get out of these assays. And so you can see on the x-axis, each letter represents different kind of water associated with the wastewater treatment process. Each sample is run in a 4-series solution. And you can see this is data for estrogen receptor alpha. And really, the take-home message is you get different hits and different types of treated water as it goes through the wastewater pipeline. In this case, we see positive hits for effluent 2, effluent 1, membrane filtration processes, and then this is just another unrelated sample here I'm not going to talk about. And then you can start comparing these to what we see with, there we go, with the androgen receptor, which we didn't hardly see any hits at all. You see that there's just a positive hit for effluent 2. Glucocorticoid receptor was also run, which really parallels what we saw with the estrogen receptor alpha. So how is such data potentially useful? And first, it provides an indication of overall extract activity from which one can calculate and quantify response thresholds like EC50s and provide benchmark effect concentrations. So such information is valuable for guiding tiered frameworks, and I'll come back to that point in discussion in a few minutes. This paper was interesting also because it recommended an ideal battery of bioassays to use for complex wastewater treatment effluents, which are highlighted in this clustering diagram. And really the take-home message here is you have the different types of water along the top and the different assays along the vertical axis here. And where we see red and yellow is where there were positive hits, and you can see that those are related to things like induction of xenobiotic metabolism, endocrine disruption, and reactive modes of action, and so forth. Ideally, what we want in the in vitro assay, however, is to be predictive of some sort of adverse outcome in vivo, especially when we're dealing with complex samples, which brings me to the case of study 2. And the goal of this work is done by Nancy Denslow, was to determine whether there could be a quantitative link between in vitro screening of compounds using these types of assays and key cellular and tissue outcomes. The study was designed around an existing AOP knowledge and FISH, which includes activation of estrogen receptors, induction of egg yolk genes under the control of estrogen that we know very well, so vitellogenin is one of these, and a response of sex ratio. They used a model of Menidia, an estrogen species commonly found along the east coast of North America at 2 life stages. And chemicals used were estradiol, estrione, and nonylphenol. So they also used this in context with the gene laser estrogen receptor alpha transactivation in vitro screening assays. And so I'm just going to really talk about a little bit of data just as a proof of concept from the juvenile study, which was a 28-day exposure and varied concentrations to these known estrogens, which were also alluded to earlier in some of the talks. And this slide really summarizes the main results. On the top left graph, the results of the in vitro estrogen receptor alpha assays show expected dose response curves, where the farthest to the left is estradiol and the farthest to the right is nonylphenol, being a weak estrogen. That makes sense. So now if you look at, this is sort of covered up here for some reason, underneath the sex ratio are gene expression changes, but it holds the same story. What you actually can see in, there we go. What you can actually see with respect to both is that the calculated EC50 based on the in vitro assays really is above what we see with both the sex ratio and the gene expression. And so sort of the point here is shown in this schematic. If you look at the data together, you can see that there's a close relationship between this in vitro data graphed here from the screening assays, which is the blue line representing estrogen equivalency, and then the other lines which represent sort of the gene expression in this context. And in fact, the in vitro assay shifted a little bit to the left, more sensitive by approximately tenfold. For the juveniles, you can see that the sex ratio is also graphed on here. But again, the take-home message is if you're using, say, the EC50 for a threshold, you are below the place or the threshold where you would actually see in vivo effects. So it's based on the information shared from these case studies, it's clear we're making great strides in inroads and moving towards utility of these assays for monitoring complex environmental samples. But there's several areas where you need to focus efforts, such assays, so they can be considered to inform regulatory practices. And so one can think about bioanalytical assays being integrated in some kind of tiered framework, say, for example, a wastewater treatment plant, whereby a certain level of activity triggers a decision matrix that may include no further testing, additional in vitro testing, or animal testing. However, to be able to use this appropriately, we need to decide on the best way to standardize methods, considering aspects such as dose addition models, work that Vicki Wilson Gray and others at the EPA and NIEHS have been focused on recently. We need to better understand the linkage between complex mixtures and in vivo adverse outcomes, and to find most appropriate suite of bioassays to make sure they are commercially available. Otherwise, regulatory use down the road cannot be enforced. One way to try and prioritize a suite of assays is to look at occurrence of activity with determined predicted no-effect concentrations. We also need to remind ourselves that while we understand what type of chemicals are associated with biological activity singly, and perhaps in some mixtures, there still exists unknown unknowns, which lead me into my second grouping of points for discussion. This is where there's a role for non-target chemical analysis of micropollutants, which I'm proposing be performed in tandem with in vitro assays. It's a field which is evolving to combat the bottleneck of our ability to identify chemicals in complex water samples with confidence. Performing such analysis will help to define the relationship between chemical mixtures and bioactivity and support effects-directed analysis, where levels of bioanalytical measures can be associated with select chemical fractions. And finally, enhancing our chemical database and environmental samples can inform on new and perhaps better directed assays for chemicals we have not yet focused on, such as neurotoxins or immune-based compounds. And so just as a brief description of how we're trying to get at this a little bit, and this is a comprehensive workflow for organic micropollutant identification. So this is consider non-target analysis where you're trying to screen for everything chemically in a sample. And so we're working with Lee Ferguson at Duke University, who has developed a complicated pipeline for identifying micropollutants in complex water samples, which is shown in the schematic. And I'm not going to actually go through all of these different iterations here of how this works, but essentially you take mass spec raw data files and put them through a variety of different programs to be able to postulate structures. And so you're essentially doing in silico fragmentation to try to identify compounds in a complex mixture. And an example of how we're using this is presented here with work we performed at Kiawah Island, which is a small island located just off the coast of South Carolina. The island is primarily home to retirement communities situated on golf course greens. They have their own wastewater treatment plant and use treated wastewater to irrigate the turf, which is also subject to pesticide and herbicide applications. Runoff from the greens goes into these retention ponds, and then these ponds are connected through an underground network of culverts that eventually make their way to the ocean. And this map shows several sampling sites, pond 25, pond 5, and pond 43, which served as our reference point since it didn't receive turf runoff or irrigation from the wastewater. And so, just as I mentioned earlier, we ran some of these, hormonally active-based in vitro assays. This is for estrogen receptor alpha on the left and progesterone receptor on the right. And you can kind of see how we utilize these assays to come up with BEQ values, but basically looking at your alpha activity, you can see the order of the greatest to the least, if you will, for both of these assays. Then we did our typical targeted micropollutant analysis, and there's lots of things present in there, and these red values actually correspond to nanogram per POSIS sampler that we put out in the ponds, and so there's some of the usual suspects there, and with a constant focus on the estrogenic ones, you can see down here that several estrogenic compounds were found in the wastewater treatment effluent, but really didn't sort of add up to the effects that we saw just based on those alone. But using a non-targeted workflow, Ferguson's group was able to come up with an additional almost 3,000 features that we're currently working on identifying. And so here we show a heat map of all the detected features with log transform responses scaled by row, sorted by sample site and date down here on the bottom, and sort of on the y-axis here, the feature category, whether they're typically found in wastewater, turf grass, and stormwater. And the colors dictate whether levels were high or levels were low. But really the point is, using this sort of methodology, we're able to start to, and this is not comprehensive at all and not published, several pharmaceuticals in this case that were never detected or were presented in the literature to be detected in the environment at all. Some that had been detected previously, and you can see these are a lot of pharmaceuticals related to beta blockers and high blood pressure and things like anticonvulsants. And so I wanted just to mention one more point briefly that might be useful for the discussion, is the incorporation of omic data as a link to sensitive molecular initiating events. Just as non-target chemistry can form on the development thresholds for in vitro assays, so can omics allow for expansion and in some cases refinement of molecular initiating events and adverse outcomes. So we should consider not just transcriptomics, but evolving omics in aquatic systems, as Stan mentioned earlier, to cover more biological space. And so we're using some of these omics methodologies in some of our current work. And so just as proof of concept, this is the brain phosphoproteome of fathead minnows exposed to two hormonal agents, ethinyl estradiol and levonorgestrel, which are two components of the birth control pill. And this graph really just shows the different changes in phosphorylation sites specific to different proteins. And so EE2 is red and levonorgestrel is green. And the idea is if the line goes up, it's increased phosphorylation event. If the line goes down below zero, it's a decreased phosphorylation event. And what you can clearly see is there's certain phosphorylation events specific to each one of these compounds related to different parts of or different processes such as neurogenesis and differentiation and vesicle-mediated transport, calcium export in the levonorgestrel exposure. And so it's such data on signaling should be considered, and perhaps events such as phosphorylation can be considered as new molecular vitiating events. And so with all of this data, I think we have to think about how this can be incorporated in these AOPs and help direct tier-based in vitro screens. And so I think important areas of discussion relate to sort of three areas: thresholds to minimize false negatives, tandem advanced chemical analysis, and new omics that can help direct incorporation of in vitro assays into monitoring strategies. There could be some very interesting discussion about trust and confidence related to the type of work presented here, and consideration of complex environmental samples and such dialogue. And lastly, I'd just like to acknowledge all the folks who contributed in some way to the work I talked about today. Thanks.
Can you hear me? There it is. Great. Thank you, Tara. Probably have time for maybe one or two questions. Anybody has one? Oh, okay, Gary? Oh, and just a reminder, can you just announce yourself for us?
Yeah, this is Gary Ginsberg, Connecticut Department of Public Health. Tara, thank you for your resiliency in stepping up and recreating your talk after the adversity of the last couple of days. So, number one, thanks for that. And for all the really interesting work you're doing in water quality. Can you talk a little bit about what kinds of decisions some of this research may be able to inform regarding the use of water or the treatment of water to make it more usable for human purposes?
Excuse me. Yeah, I think there's two areas that we're interested in has importance in public health arena, and that certainly is monitoring of waste water in general, right? So the idea is, I think, if in our minds, and we've had discussions about this, can you come up with a suite of assays that make sense in monitoring for things like spikes that might come through a wastewater treatment plant? And the second is, of course, water reuse, because there's a big push for that. And Nancy Denslow, unfortunately she's not here, could speak to this better than I can, but she's highly involved with folks out in California who are really trying to push the use of these for water reuse and eventually water reuse for drinking water purposes, right? And so to be able to come up with some tiered screening system to be able to use reuse water for drinking water or potable standards. And so there's a lot of discussion about how best to do that. And, I mentioned thresholds, but I think in the EU, for example, I think there'll be some talk about this tomorrow, perhaps, that I don't think thresholds are considered as much, so is that the way we should sort of go for these complex water samples or not? So I think there's still a lot of debate and discussion about how best to do that.
All right, great. Thank you.